We typically isolate neurodegeneration into independent silos: synapses failing from excitotoxicity, mitochondria collapsing from NAD+ depletion, and the hippocampus losing adult neurogenesis. But approaching these as separate failures creates a blind spot. They rely on a shared metabolic infrastructure, drawing from the same raw material supply. We can map the physical deterioration of the brain across 3 specific axes: the decay of the synaptic circuit, the breakdown of the microglial state, and the exhaustion of the bioenergetic substrate. These 3 axes of collapse are not merely correlated phenomena. They are actively coupled together, gated by a single metabolic bottleneck. That bottleneck is a specific essential amino acid, L-tryptophan. Out of all the amino acids required for mammalian life, tryptophan is the rarest. It makes up only about 1% of our entire proteome, and its concentration in human blood plasma hovers at an exceptionally low 50 to 80 micromolar. In this animated model, we can see how the brain operates a central tryptophan pool that must simultaneously feed 4 distinct biochemical branches: general protein synthesis, the kynurenine energy pathway, the serotonin neurotransmitter pathway, and the microbial tryptamine branch. The annotation of tryptophan across these 4 branches is a highly competitive zero-sum fluid dynamics problem. When one branch demands more substrate, it physically drains the supply available to the other 3. We can observe the baseline allocation of a healthy brain using a Sankey diagram. Under normal homeostatic conditions, the vast majority of the central tryptophan flux is consumed by basic protein synthesis, along with a slow, steady trickle into the kynurenine pathway processed by the liver. The remaining fractions of a percent are routed down into the tryptamine branch and the critical serotonin branch, which is governed by an enzyme called TPH2. The TPH2 enzyme requires a minimum concentration of tryptophan to function. This kinetic threshold, its Km, sits at approximately 25 micromolar. In a healthy state, the central pool hovers just slightly above this line. The central serotonin system is normally saturated and quite stable, but its margin of error is nearly non-existent. It is extraordinarily vulnerable to any drop in upstream substrate. The structure holds until it encounters a specific stressor, chronic neuroinflammation, typically driven by immune signaling molecules like interferon gamma and interleukin-1 beta. When the brain's immune cells, the microglia, become chronically activated, they initiate a defensive response. They express an enzyme called IDO1, which directly taps into the central tryptophan pool. IDO1 possesses a high catalytic capacity. It immediately routes the tryptophan flux away from normal functions and forces it down the kynurenine pathway. IDO1 is an unrestricted metabolic sink that forcefully starves the brain's survival pathways of their required chemical substrate. When we trace the effects of this IDO1 shunt back to the serotonin branch, the central tryptophan pool level drops below the 25 micromolar threshold required by the TPH2 enzyme. Because the substrate is missing, the synthesis of serotonin and the vital neurogenic signaling proteins that rely on it physically stops. For decades, the medical standard for addressing the depressive symptoms and cognitive decline of early Alzheimer's has involved prescribing SSRIs, selective serotonin reuptake inhibitors. The mechanism of an SSRI relies on keeping existing serotonin inside the synaptic cleft, but a reuptake inhibitor cannot amplify a signal that the brain lacks the raw material to synthesize in the first place. The drug fails because the substrate has already been stolen upstream. To understand the damage, we have to follow the stolen substrate into the brain. Once tryptophan is forced down the kynurenine branch, it hits a geographical fork in the road. If the kynurenine is absorbed by astrocytes, an enzyme called CAT converts it into kynurenic acid, or KYNA. This is a broad-spectrum neuroprotectant that dampens overactive neurons. Under neuroinflammatory stress, the activated microglia deploy a competing enzyme called KMO. KMO aggressively outcompetes the astrocytes, converting the accelerated kynurenine flux into quinolinic acid, or QIN. QIN is a potent competitive NMDA receptor agonist. When it accumulates in the microenvironment, it locks the neuronal receptors open, causing a surge of calcium that destroys the synapse. The inflammatory kynurenine shunt delivers a simultaneous double blow to the neural circuit. It starves the environment of the neuroprotective KYNA it needs, while flooding the vulnerable synapses with toxic quin. The immediate question is why mammalian biochemistry permits a lethal neurotoxin like quin to accumulate. The answer lies at the very end of the pathway. Quin is actually the required precursor for the de novo synthesis of the critical energy cofactor NAD+. In a failing cell, hyperactive DNA repair enzymes, specifically PARP1, are aggressively consuming the cell's baseline NAD+ reservoir. The cell is suffocating, and it forces flux down the kynurenine branch in a desperate attempt to manufacture replacement energy. But the final enzyme in this sequence, QPRT, has a strictly limited processing capacity. The surge of quin hits the bottleneck, backs up, and spills out into the surrounding tissue. The entire process is an inherently wasteful evolutionary gamble. The cell destroys its own synapses with quin toxicity, all without ever successfully resupplying the exhausted NAD pool. Because the decay of synapses, the inflammation of microglia, and the collapse of cellular energy are all coupled to this single upstream partition node, attempting to treat any one downstream symptom in isolation is mathematically futile. Rebalancing the system requires an integrated approach. First, the pathological shunts at IDO1 and KMO must be chemically inhibited, effectively closing the floodgates that route tryptophan into excitotoxic quin. Second, we must bypass the QPRT bottleneck entirely by introducing direct NAD+ precursors alongside raw tryptophan loading, refilling the drained central pools. With the shunts closed and the substrate restored, the entire metabolic system repressurizes. The toxic quin flood recedes, the TPH2 enzyme hits its required threshold to resume serotonin synthesis, and the cell regains its bioenergetic baseline. Correcting the tryptophan partition addresses the underlying fluid dynamics problem, halting the bioenergetic, microglial, and synaptic collapse simultaneously. We have historical proof that correcting the specific metabolic failure restores cognitive function. It is found in pellagra, the original disease of tryptophan and NAD+ partition failure. Pellagra is characterized by 3 severe symptoms: dermatitis, diarrhea, and profound dementia. In the early 20th century, researchers identified that the severe cognitive decline caused by pellagra was entirely reversible. By simply supplementing patients with raw tryptophan or niacin, the missing metabolic substrate was restored, and the dementia cleared. The bioenergetic starvation observed in early-stage neurodegeneration mirrors the mechanics of pellagra. The neurons are struggling for substrate. Alzheimer's and related dementias feature a substrate-correctable state early in their pathology. If we rebalance the metabolic partition before total structural collapse occurs, the physical deterioration of the brain can be halted.